Richards obtained her Bachelor in Science in 1990 and her Doctorate of Philosophy degree in 1994 at the University of Melbourne in Australia. She is Professor of Neuroscience and Deputy Director of Research at the Queensland Brain Institute at the University of Queensland, Brisbane, Australia. Linda is Fellow of the Australian Academy of Science and the Australian Academy of Health and Medical Science, is a National Health and Medical Research Council Principal Research Fellow, and was appointed an Officer of the Order of Australia in 2019. She is past President of the Australian Neuroscience Society and co-chair of the Australian Brain Alliance. In October 2018, she was appointed to the Federal Government Million Minds Mission Advisory Panel. Linda is head of the Brain Development and Disorders Laboratory at Queensland Brain Institute. She is leading expert on the formation of the corpus callosum and is scientific advisor and patron for the Australian disorders of the corpus callosum. Her laboratory team strives to understand how the brain forms during development and how these processes are disrupted, causing human development brain disorders and brain cancer. Linda has a prolific bibliographic production with more than 125 research articles, book chapters, and books. Linda has received a number of awards and fellowships throughout her career, including the Charles Hudson Herrick Award and the American Association of Anatomists in 2004, and the Nina Condelos Award from the Australian Neuroscience Society in 2010. In 2015, she co-founded the International Consortium for the Corpus Callosum and Cerebral Connectivity that bring together clinicians, scientists working in different cases of developmental brain disorders and how best to provide support and care for affected individuals and their families. Professor Richards is passionate about public awareness of science, and in 2006 she founded the Australian Brain Bee Challenge, a competition for high school students to learn more about the brain. So sorry for this difficult start, Linda, and thank you very much for being with us. The screen is yours, and we are going to show you a presentation. Thank you, Linda.
Thank you very much, Marita. You can hear me okay?
Yes, perfectly well.
Terrific. Well, good morning it is for me. I imagine it's good afternoon or good evening for all of you. And it's so exciting to be able to present across the world to you and to tell you a little bit about our work today. I will try to leave lots of time for discussion and questions at the end. So we can start. What you're seeing here actually is a technique that we use in our laboratory, which is based on MRI diffusion imaging. And this is a form of tractography that we use, where these colored lines are actually streamlines that are generated by the computer that are color-coded based on the axon tracks within the brain and the orientation of those axon tracks. So those tracks that are running from left to right are colored in red, and those that are running from front to back are colored in green, and the descending projections are in blue. So I'll come back to that a little bit later. So our laboratory is very interested in how the brain is formed and wired up to function, and the function part is very important. We want to understand how the wiring of the brain and the major axon tracks in the brain underpin brain function. So we're looking at processes that occur early in development to build the wiring of the brain and how these impact cognitive function. And the major, as Marita kindly said in her introduction, the major fibre tract in the brain that we study is the corpus callosum, which is this huge fibre tract that connects the two cerebral hemispheres together. Interestingly, the corpus callosum is only in placental mammals, and did not evolve in marsupials and monotremes. So this is only found in placental mammals. And we do work with Australian family support group, Australian Disorders of the Corpus Callosum, and we're interested in how these disorders occur and how they affect brain function. So we have a group of research participants that we work with, and when they come into our study, we are performing genetic studies with our collaborators in the US and other places around the world. And we perform multimodal MRI imaging on these participants and also neuropsychological testing, again, in collaboration with Gail Robinson, whom I work with here. And then we take this information and we use animal models to test our hypotheses and try and get at the mechanisms. So this group of disorders, midline disorders of the corpus callosum, occur in around 1 in 3,000 live births and is associated with a large number of different congenital syndromes. So this is where the person has a corpus callosum disorder, but also another anomaly. So it could be another brain anomaly or another organ system anomaly, maybe of the heart, the kidneys, lungs, etc. But there are people who have only corpus callosum disorder and a couple of associated brain malformations that always co-occur with complete agenesis of the corpus callosum, as it's called. And these people have been studied fairly extensively by a group at Caltech and the Fuller Institute in Los Angeles, so Lynn Paul and Warren Brown. And we know that these disorders, both the ones that are shown here, so these cognitive outcomes, but also the congenital syndromes also result in, can result in sensory motor abnormalities and in learning delay and learning difficulties. The people that have isolated agenesis of the corpus callosum were, as I mentioned, studied by Lynn Paul and Warren Brown to have this core syndrome of 3 specific areas. So slower cognitive processing, restricted processing of complex and novel tasks, which is really quite interesting and demonstrates some deficits in the interaction between the hemispheres, particularly in executive function and in those frontal and prefrontal areas. And of course, a limited interhemispheric transfer of complex sensory motor information, as I mentioned before. So as Marita also mentioned, we formed this international research consortium for the corpus callosum and cerebral connectivity in 2015 with colleagues from all around the world. Some of you may know Professor Fernanda Tovar-Moll. She's from Rio de Janeiro and is a major director of, one of the directors of our consortium. You can learn more about our consortium at our website. So there are different types of corpus callosum malformation. So I mentioned briefly before complete agenesis, where the axons fail to cross the midline, but there are also people with partial agenesis, or this small callosal remnant that remains, and actually everything in between that small remnant and a fully formed corpus callosum. So different corpus callosum of different sizes. And corpus callosum can also be thinner or thicker. And thinner and thicker are also lead to or associated with cognitive deficits. So as I mentioned before, these are the tractography images, and we're looking down on top of the corpus callosum now in the neurotypical case. And in this case, with a person with complete agenesis of the corpus callosum, you can see that the fibres, instead of crossing the midline, they actually form these longitudinal bundles, which are known as Probst bundles because they were discovered by Probst in 1901. And you can see that in people with this partial ACC, that there are some axons crossing the midline, but there's also a lot still forming these major longitudinal bundles. And both Fernandez's group and also Elliott Scher's group in San Francisco have found that some of these individuals with partial agenesis of the corpus callosum have a completely ectopic tract known as a sigmoid bundle. And this sigmoid bundle is not normally seen in neurotypical individuals and connects the right frontal lobes with the left occipital lobes, and it's often asymmetric. So you can find this projection going the other way, but it's usually larger in one direction. So what this tells us is that brain wiring is not the same in everyone, and that there are brain wiring variations that can be revealed by studying people with corpus callosum dysgenesis. And so this introduces another concept, which is this idea of long-range axonal plasticity. So we're all familiar with synaptic plasticity that occurs throughout life. I'm talking about long-range axonal plasticity, whereby during brain development, axons may take alternative routes in order to wire up the brain. So we're interested in the mechanisms, how do they do this, and also how do these alternative routes underpin brain function. So here are some images. This is quite a busy slide, but I'll walk you through it. So here are some images from our cohort in Brisbane. So these are 7 of our research participants that have partial agenesis of the corpus callosum. So here is our example of our neurotypical controls. And in this image here, what we've done is taken the sagittal view and just traced around the callosal remnant that remains so that you can see the variations. And then on this side, what we have is the tractography that Tim Edwards and Ryan Dean in my group have performed. And what you'll notice just looking at a very gross level is how different the connectivity is between these individuals. And so some have more frontal connections than others. And this has also been seen by other groups, Elliott Scher's group, as well as Fernandez's group in Brazil. And so even when the partial remnant is a fairly similar size and located in a fairly similar location from rostral to caudal, the actual connectivity through that remnant is quite different between different individuals. So we find this really interesting, and we'd like to understand how the brain is wired up in these individuals. So we turned to using an animal model to try to get at some of this. So this is Tim Edwards, who did his MD PhD in my group, and he was co-supervised by my collaborator at UCSF, Professor Elliott Scher. So what Tim did was to take one mouse strain, which is called the BTBR mouse strain, available from Jackson Labs, that has complete agenesis of the corpus callosum, and he crossed that with a C57 Black 6 normal laboratory strain. And when you do this, you get an N1 generation, which you can then cross back to the BTBR strain. And the pups from this N2 generation have variations. Even within the same litter, there are animals with complete agenesis of the corpus callosum, partial agenesis, and full corpus callosum. So this is a way that we can get control litter mates and animals within the same litter that have varying corpus callosum. And then what Tim can do is to perform MRI tractography but also, of course, in mice, we can perform tract tracing. So these are just MRI color maps, just showing the different littermates. And you can see these are the group of mice that were studied by Tim, some with very small corpus callosum and some with almost normal corpus callosum. So this is the great range of different partial agenesis that you can see compared to an animal with a complete or normal corpus callosum. And so what Tim actually wanted to do was to see whether mice have this sigmoid bundle as well, this ectopic bundle that we had seen in human subjects from the work of Fernand Tovar-Moll and Elliot Scher's groups. And indeed, what he found was that mice do have a sigmoid bundle, and interestingly, it is also asymmetric. So much thicker, more axons traveling from right to left than from left to right. And he teamed up with Laura Fenlon, a postdoc in our group, to actually trace these axons using a technique called in utero electroporation. So we can introduce a plasmid that is expressing TD tomato, in this case, into this lateral region of the cortex. And normally these axons are, so you can see here in the control, the corpus callosum connects homatopic regions in the contralateral hemisphere. So what you're seeing here is a horizontal mouse brain section, and the embryos were electroporated during development, and then the mice were allowed to grow up, and then we imaged their brain, and then we sectioned them. And what you can see is if you compare where a sigmoid bundle might be. So this is a mouse with partial agenesis of the corpus callosum compared to the animal with a full corpus callosum. You can see that these different region of interest, 1 and 2, are quite different between the control and the animal with partial agenesis. So what we see is an increase in axonal numbers that are going into this rostral region in the partial agenesis compared to the control. And we see a corresponding decrease in the amount of axons that are growing here. So instead of targeting their normal target, these axons are targeting this rostral region. So we don't know why they do this, or even what are the mechanisms, the molecular mechanisms by which they do this, but this does give us a model to study this. Now, Tim also applied a different technique, which is called graph theory analysis, to his structural tractography data. And this is a technique that has been applied in humans as well. And these are Tim's mice with the controls, so the full corpus callosum, complete agenesis, partial agenesis of the corpus callosum, and then this is a different experiment that he did where he removed the fibres that would normally be crossing the midline here in the control animals, and this is a virtual callosotomy. So what you can see are these round dots here, which represent nodes throughout the brain of major connectivity. So the size of the dot represents how much connectivity is coming into that area. So remember, this is structural MRI, not functional MRI. Sometimes you see these maps also for functional networks, but this was these brains were image fixed. And then in these circular maps, what you can see are these are different regions of the brain in the left and right hemispheres. So you can get a sort of circular map of connectivity as well. So just two different ways of looking at the same data. And what you can see is actually, it's not just that when you remove the corpus callosum, either in an animal with complete agenesis or using a virtual callosotomy, that you just take out some of these axons, and that's all that happens. What seems to happen, in fact, is that you get a reorganization of the connectivity in each hemisphere in these congenitally acallosal mice. So having no corpus callosum is one thing, but it also impacts how the axons are connected throughout each hemisphere, and the size of the nodes in each hemisphere again are indicating this reorganization. And it also demonstrated just how different animals with complete agenesis of the corpus callosum and partial agenesis of the corpus callosum are different as well in terms of their connectivity. So all of this is really to say that all brains are definitely not wired up the same, and that there are major differences that occur that can be revealed by studying these disorders of the corpus callosum. So, of course, we're very interested in how does this impact function? Does it impact function, or can you get the same function with this reorganized set of connections? And these are really open questions at the moment. But also this work identifies perhaps two important points regarding callosal wiring disorders. So at the moment, people are diagnosed as having a corpus callosum disorder at the midline. So this is where all the axons arrive at the midline and are crossing. And this is how they are really diagnosed. But the axons are actually coming from mostly layer 2, 3 neurons, but also layers 5 and 6 have major projections across the corpus callosum. And so these neurons are, these neurons are sending out their axons towards the midline and then usually projecting into the homotopic area in the contralateral region. But it's possible, given our evidence from studying acallosal brains, that in fact, even in brains that may look like they have a normal corpus callosum, that there could be abnormal contralateral targeting where the midline crossing is normal, but actually there could be targeting to the wrong area, for example. And so it's something that we are interested in. We don't have a lot of evidence of this now, but we do have some evidence in people and some evidence from animal models that certainly axons are very malleable and plastic in where they might project. And we need to really understand how important this point-to-point connectivity is in terms of function. So I just wanted to now switch to talk a little bit about how one might see different brain abnormalities in the same individuals that are associated with corpus callosum malformations, and whether this can give us some kind of handle on early brain developmental processes that give rise to multiple disorders in the same person. So we've been studying a number of genes, and I'm going to talk about two of them here, the nuclear factor 1 gene family and also a gene called deleted in colorectal cancer. And I want to talk about how these are involved in both corpus callosum agenesis, but also in conditions such as ventriculomegaly, so enlarged ventricles, and enlarged brain, which often leads to intellectual disability. So, right. So let me start firstly with the corpus callosum anomalies. And this is work that was conducted by Dr. Ilan Gobius when she was a postdoc in my group, together with Laura Morecambe, who was a student at the time. So they set out to try and understand what is the cause of complete agenesis of the corpus callosum. And the key here are these two small pieces of tissue that sit at the midline known as the septum pellucidum. And you can see that in neurotypical individuals, these septum, two sides of the septum are fused together. But in people with complete agenesis of the corpus callosum, they also display these open septal leaves. And this is really a key to understanding how agenesis of the corpus callosum actually occurs. So just before I go into some more details about that, a little bit more on background, and that is this concept that glia, the other very, very important cell type in the brain, are very important for wiring up the brain and the nervous system of all animals and humans. So what you can see here is the midline of, this is a mouse section at postnatal day 10, focusing in on the midline and staining with glial fibrillary acidic protein in green. And these axons, the callosal axons, have been labeled with dye-I. And here is where we started to first really get interested in this family of genes called the nuclear factor 1 genes, because they're very important for glial development. Now, we've also in the past, our group and many other groups in the field have been studying mechanisms of axonal guidance. So this is where callosal axons and axons of all types in the nervous system, when they're wiring up the brain, they express receptors on their growth cones and use the environment and ligands such as these, with names like SLIT, semaphorin, and netrin, to navigate their way to their correct targets in the brain. So I'm going to come back to DCC a little bit later, but I'd now like to talk about the nuclear factor 1 genes. So these are a family of transcription factors with 4 family members, and 3 of these family members are expressed in brain. They each have the same DNA consensus binding domain, and this is work done by all of our collaborators, and in particular, I'd like to highlight Professor Richard Gronostowski, who we've been working with on these genes for around 2 decades now. These genes are important because they sit at this interface between proliferation and differentiation. So early in development, they seem to regulate this switch, turning off proliferation and turning on differentiation. And I'm highlighting the glial cells here, but they're also expressed in neurons. Now, Rich Gronostowski has made knockout mice of each of these, and these mice have severe cortical phenotypes, which include dysgenesis of the corpus callosum, hippocampal malformations, and ventriculomegaly. In addition, we, together with collaborators overseas, have been studying human NF1 developmental disorders. And these are, so there are people with that have mutations in NF1A, B, and X, and they have some overlapping brain phenotypes: megalencephaly, enlarged brain, ventriculomegaly, dysgenesis of the corpus callosum, and developmental delay. So in this paper, we analysed different human mutations throughout the nuclear factor 1B gene, and have assessed sort of how these genes might function. So we know also, here's an older study from 2007 in a nuclear factor 1A patients, showing a variety of different callosal anomalies as well. And so just going back to the cause of complete agenesis of the corpus callosum, together with Elliot Scher's group, we looked at 38 different cases and tried to see, was this always correlated with these open septal leaves or not? And it turns out that complete agenesis is pretty much always correlated with open septal leaves. So how does this occur? How do you get these open septal leaves? So during early development, when the cortex is forming from the prosencephalon to form the 2 telencephalic hemispheres, You can see that the two hemispheres are actually split, of course, because they're ballooning out. They're connected ventrally, but in this more dorsal or middle area here where the callosal axons would cross, and this is a scanning EM of a mouse embryo at embryonic day 12, and these are pictures of human gestational week 12 brains, just showing this interhemispheric fissure. And if we take a human gestational week 13 brain and look at it in the scanner, this is a fixed brain, we can see that in fact the septum does have various, even within the same brain, areas where it appears to be unfused and areas where it's fused. And when we section these brains at 13 weeks, 14 weeks, and 15 weeks, you can see we stained these sections with GFAP, glial fibrillary acidic protein, to label the glia. And we could observe in human brain and in mouse brain these midline zipper glia that were first described by Jerry Silver and colleagues that line the midline here and initially start out as bipolar cells, which then become multipolar astrocytes and fuse the midline together in this kind of a zipper-like fashion. And here you can see them in mice at E16 and E17 before midline remodeling with this bipolar morphology, and then right at the midline once they become intercalated. So this is mouse developmental stages, and you can see the midline fissure, the interhemispheric fissure, actually fusing here, and over time by these glia, which are labelled with nestin. And essentially, these glia provide a substrate for callosal axons to cross the midline. And here we've labelled the very first pioneering axons, and you can see them approaching and crossing the midline. This is mediated, so the development of these midline zipper glia is mediated through this morphogen, FGF8, and via the receptors FGFR1 and FGFR2. And we know that FGF mediates this differentiation of the midline zipper glia via the nuclear factor 1 genes, because when we overexpress FGF8, we can induce the expression of GLAST and then GFAP. But if we do this in either an NF1A or B knockout, we cannot induce this. And we also find patients with NF1A or B haploinsufficiency have this open interhemispheric fissure in the same way that the mice do. And so this is the model that we published some time ago, showing that the interhemispheric remodeling event actually provides a substrate for callosal axons to cross the midline. And incidentally, this does not occur in marsupials and monotremes. So we have this paper demonstrating that both this little fat-tailed donut marsupial that we study in our group and the platypus failed to undergo this interhemispheric remodeling, suggesting that it is an essential part of a formation of a corpus callosum, because remember, these animals don't have a corpus callosum. So I was just briefly going to touch upon DCC, but I might run through this fairly quickly in the interest of time, Marita. So DCC is a transmembrane protein deleted in colorectal cancer. So it's a classic axon guidance receptor that has been shown to bind a variety of ligands, most notably netrin-1. And it interacts with these intracellular signaling molecules to regulate the actin cytoskeleton. And a number of years ago, we were studying DCC knockout mice, and we noticed that they had complete agenesis of the corpus callosum. And this was first published actually by Serafini et al. And Mark Tessier-Levine's group. that these mice had complete agenesis of the corpus callosum, and they do have axonal guidance defects at the midline, but you can see that they also have an interhemispheric fusion defect. And this was interesting because patients with DCC mutations were first identified by Guy Rouleau's lab in Canada and Emmanuel Rose's group in France. And they found that people with DCC mutations had a condition, which I'll show you in a moment, called congenital mirror movement syndrome, but they had normal corpus callosum, or normal midline corpus callosum at least. And so this was perplexing for us because we had mice with knockout of DCC that had complete agenesis of the corpus callosum, but human studies showing that these patients had a normal corpus callosum. And so it wasn't until recently, so we published this paper together with the group's back-to-back paper from Chris Walsh, Elizabeth Engel, and Timothy Yu's groups, showing that there are patients actually that do have callosal malformations that have DCC mutations. So it was really an example of where there just hadn't been enough patients actually studied at that time. And now there are a large number of human mutations that have been identified, and this is even a little bit out of date now, in many areas of the DCC protein. And these different mutations do seem to have an effect on the outcome and the function of the protein. So here is a child that has this congenital mirror movement syndrome, and what it is that he's being asked to move one hand, but he's unable to inhibit the other hand from doing the exact same movement. And these mirror movements can happen in the feet as well as the hands. So it's not a life-threatening disorder, but it is actually a way of diagnosing these patients. And patients that have mirror movement disorder have been associated with a large number of different genes, or these 4 genes at least now as well. We were interested in how DCC might be functioning in terms of interhemispheric fusion. So we had the netrin knockout mice. These are actually a hypermorph because it's a beta-gal knocked into the netrin gene. And the DCC knockout mice, and then another animal that we've been studying called DCC kanga, which has a DCC protein but lacks this P3 domain, which is very important for interacting with the actin cytoskeleton. And you can see that each of these knockouts, the ligand and the 2 different DCC knockouts, have an open interhemispheric fissure. And here is some actual sections showing you where DCC is expressed, and in these cells that give rise to the midline zipperglia here, and also that netrin is expressed in this area as well. And what we find is, so this is the DCC mutant and the netrin mutant, is that these progenitors cells that form in this area where the midline zipperglia form, so this is a horizontal section, and this is actually the lamina terminalis, the floor of the 3rd ventricle, in a kind of hinge area. It's greatly reduced in both of these mutants. And in fact, we see some alterations in the migration of these cells forward. These are horizontal sections. And this has actually been posted on bioRxiv. So if you want to have a look at the details, you can. We also studied the kanga mutation in cell lines, and we find that the kanga mutation actually disrupts this interaction with the actin cytoskeleton. And we started to look at some of these human mutations to see whether they might also disrupt this function of DCC. And so here, when you overexpress wild type DCC, you get a lot of finger-like projections in the cells. And in fact, that's disrupted in the cells that have the mutant introduced into them. So we think that not only is FGF8 and MAP and ERK pathway regulating NF1 genes that regulate these midline zipperglia, but also that netrin and DCC are required for the formation of these midline zipperglia as well. I could stop there, Marita, if you would like, or run through the role of NF1s in ventriculomegaly.
I think you can continue, Linda. Yes, of course.
Okay.
Yes. We are 10 minutes to 7, and we started a little later than 6. So
okay.
please go on. Thank you.
Keep going then. So just going back here. So I'd just like to talk a little bit about how these genes are not only involved in the actual formation of the substrate to form the corpus callosum, but actually also because they regulate radial glia development, because remember that these midline zipperglia arise from radial glia in the floor of the third ventricle. And so this actually connects very closely with the other phenotypes that are seen in these patients with NF1 mutations that have enlarged head and brain and enlarged ventricles. So here, just some of the evidence of the enlarged ventricles in NF1A or NF1B mutations. And in fact, this one is just worth pointing out because it is a very, very common phenotype in people with complete agenesis of the corpus callosum. It's enlargement specifically of the posterior horns of the lateral ventricles. So it's a condition called colpocephaly, and it's always associated with complete agenesis of the corpus callosum, or very, very commonly associated anyway. And it's interesting, it's, we believe, not associated with hydrocephalus per se, because it is really confined to the posterior horns of the lateral ventricles and not throughout the ventricular system. Of course, you can have callosal anomalies with hydrocephalus, but this is a little bit different, we think, because it's so confined. So I'll just quickly run through the basis for this, for our hypothesis. So in early development in mouse, the neuroepithelial cells divide profusely. They give rise to radial glia, which can be identified by a transcription factor called PAX6. And these radial glia proliferate and they expand the ventricular zone laterally as they do so. And that's this lateral expansion that I'm talking about here. And then at a particular stage during development, these radial glia, which have been proliferating, begin to form one cell that will keep proliferating, and another cell which will actually begin to form differentiated neurons or glia. And it's this switch from proliferation to differentiation that we think nuclear factor 1 genes are very crucially important for. So once this radial expansion has reached a certain point, then there will be a tangential expansion of the cerebral cortex as cells that are derived from the intermediate progenitors form neurons and astrocytes. And this is all work done by investigators in the field over the last 100 years. So it's really fairly well established. So this is this radial expansion that I'm talking about here. So in NF1, I'm just showing the B knockout here. In NF1B knockouts at embryonic day 14, things look very similar to their wild type littermates. But by E16, what we observed was that the lateral expansion actually continues. And this is at the expense of this radial expansion. And so by E18, just before birth, we see that, in fact, the ventricles remain larger, or the ventricular zone remains larger here than it does in the controls, but the thickness of the cortex is thinner. And as I mentioned before, that we think that NF1s operate in this transition from proliferation to differentiation, because in these knockouts, so this is PAX6, you can see that there's still a large number of progenitors in the ventricular zone of these mice, but fewer differentiated glia and neurons. And in fact, when we make NF1A and B double knockout mice, we see an even more extreme phenotype where the ventricular zone expands so much that it's actually beginning to make these folds in order to fit in the skull. And the cortical plate is really very, very thin. So this is not hydrocephalus. This expansion of the ventricular zone is due to more radial glia being added rather than hydrocephalus. And so when we knock out NF1, we get an expansion, a lateral expansion, which is even more pronounced with double knockout mice. And we then were fortunate to obtain some conditional knockout mice from Richard Gronostowski, which we then crossed with EMX1 Cree and also TD tomato flock stop mice, so we can see where the Cree is active. And that's why you can see the red here in the conditional knockout mice, because we wanted to see, does this actually result in a larger brain, which is what these patients have. And in fact, so these are postnatal day animals. And in fact, it does result in a larger cortical length and width, which persists well into adulthood. In addition, we studied their behaviour, and this is fairly preliminary and unpublished data, but we used this task. So we put them through a battery of different tasks, studying their motor abilities, their senses, and various learning and memory tasks. And for the most part, they were normal. In this task, however, it's called the active place avoidance task, where we place the animals onto a rotating platform which has a shock zone that they need to remember to avoid. So they get placed on this for 1 to 5 days, and then there's a break for 2 weeks, and then they're tested again. And what we find in the NF1B conditional knockouts is that they essentially they're poorer at this task. So they have some learning and memory deficits. And this is work that we did in collaboration with Tom Byrne at Queensland Brain Institute. So this is our evidence thus far that disruption in radial glial development, particularly through this NF1 genes, results in both this lack of interhemispheric remodeling and also changes to the brain, which begin as enlarged ventricles with actually more radial glial cells that eventually give rise to more neurons in the brain and a larger brain. And I'll just finish there by thanking the members of my group. These are the members of my current group who are working across different animal model systems, the fat-tailed Dunner, the mouse, and in our human subjects. And just to let you know that I will be moving to Washington University in St. Louis in January next year. Thank you very much.
If anybody wants to make a question, or yes. Shall I? Yes, are you unmuting yourself? Okay. Would you like to make any questions or comments?
Yes.
It was okay.
Thank you, Marita, and thank you very much, Dr. Richard. It was a very nice, so kind, and very, very understandable talk. Thank you very much. Okay, this is not my field of research. For that reason, I don't know how long the humans and the rats in your model can stay alive and what others diseases they can get?
Yeah, a very good question. So firstly, the agenesis of the corpus callosum is not lethal at all. So people live a normal lifespan, and the mice live a normal lifespan. So we think that this is really because the corpus callosum evolved in placental mammals. So it's for higher cognitive kind of processing. It's not required for like the brainstem areas for breathing, or like the hypothalamus for food intake or reproduction or things like that. It's really, in fact, some of the very interesting areas are in social interactions. And so it may affect fitness, but it is certainly not, it's not lethal at all. So that was the first question. And then your second question was about whether they might get other conditions. Absolutely possible, depending on the gene that might be involved. So I mentioned that people with corpus callosum anomalies, it's a very, very broad group. Some people just have the corpus callosum and colpocephaly. Now you know what colpocephaly is. So some people just have that, but some people have a congenital syndrome, which causes the same gene can cause different organ system problems. And so definitely they can have other things. Yeah.
Thank you very much.
Thank you for the
Thank
question.
you, Zulma. Now there is just a comment from Diana Jerusalinsky. You might remember, Diana, from last year when you were in Montevideo for the first meeting of the Lab Brain Initiative.
Memories.
Yes, and Diana says she did have to leave because she had another meeting, but wanted to thank you very much. And well, it was very happy to see you again. There is also another question from Mariana. Mariana is going to make it in Spanish, but we can translate it to English if me or the translator, if you want Mariana to make the question.
I'm not able to see the chat or the questions,
Hola.
by the way.
Hola. Hola, Mariana.
Bueno, gracias a la doctora Richards, muy interesante. Yo justamente estoy trabajando con asimetría interhemisférica estructural y quisiera saber si ustedes han explorado la implicancia que podría tener la asimetría en el haz sigmoideo del que hablaban.
Mariana says that in fact, she's very interested in your presentation because she is working in hemispheric asymmetries. Okay, and the specific question, if you have studied the implications, the consequences of the asymmetry on the sigmoid as pathway?
Yes.
Yeah.
Right. Are you asking about the cognitive outcomes for those?
Pregunta, Linda pregunta si tu pregunta concreta, Mariana, es respecto a las consecuencias cognitivas.
Sí.
O es estructura. Yes, yes. Mariana says that yes, she is asking about that.
Right.
About the cognitive consequences.
We would like to do that. So what Fernanda Tovar-Moll has been looking at some of her research participants that have this sigmoid bundle, and she finds that it is not, so they're more affected. So it doesn't seem that this is actually beneficial, this ectopic tract. But it's not very common. So she only has a few participants. And so one of the things that we actually want to do in our consortium is to pool some of these participants so we can do a study which is more harmonized across different sites, so we can have more subjects to really make some understanding of this. She has done some MEG work on them. So this is to look at activity, and she does find coactivity in the areas that are connected by the sigmoid bundle. So yeah, so it seems that it may not be beneficial for people, but it's very, very early days, so we don't have very much to go on yet.
Okay, well, Anabel Fernandez acknowledged very much your presentation. And I would like to make you a question, Linda, regarding the last factor that it was involved in the balance between proliferation and differentiation. Are you kind of trying to find a way to counteract that? So when you make the diagnosis that this is the problem, is there any possibility to kind of change this this balance between proliferation, counterbalance the genetic?
Yeah. So of course we don't have anything, and this is a transcription factor. So extremely hard to make a drug target for a transcription factor. So what we're doing is looking at the downstream genes that it might regulate, and then seeing whether any of those might be, you know, amenable for therapeutics in the future.
Okay.
So that's kind of the way we would go with that. I see Vipu is online as well. Did you want to ask a question?
Yes. Thank you very much. That was a fantastic talk. I had two sort of broad questions. First of all, I'm saying hello from New York, so I think
you're in New York. Oh, wow, exciting!
Yeah.
Wow. What is it there? It must be very late.
It's 6 in, so it's not.
Oh, okay, not too bad.
Yeah, not too bad.
Well, thank you for tuning in.
This is great.
Absolutely, an absolute pleasure. Actually, we met very briefly when I was in my postdoc with Jeff Mackless.
Okay.
And so now I've started my own lab. So I don't study the corpus callosum, unfortunately, but I'm still definitely interested in developmental mechanisms.
Yeah, sure.
And so the two sort of broad questions that I was thinking of was, I was very intrigued by the finding that the non-placental mammals in your monotremes, when you overexpress, they're not receptive to making that glass thing happen. So for some reason, there's that capability seems to have evolved more recently. So that is something that I thought was very interesting, because, you know, we keep thinking of evolutionary mechanisms and the callosal sort of projection neuron diversity has expanded through evolution, but this would argue that there's something fundamentally different on the glial side of things, such that they've sort of evolved this new capacity to build these kinds of. Are there additional kinds of such mechanisms that people have identified through evolution that are specific to the glia, and where else in the nervous system have those been seen?
Okay, really fantastic questions. Let me first start by, so actually on the screen here, you can see the little fat-tailed dunnart, and they have this enlarged anterior commissure, which essentially is like a corpus callosum with an anterior commissure sitting on top, because their entire neocortical regions are connected via that anterior commissure. And then there's also all the more ventral areas that would normally be connected via the anterior commissure are also connected via the anterior commissure. And interestingly, those anterior commissure projections are more ventral, and then the ones from the neocortex sit more on top. So Rodrigo Suarez, who's a senior scientist in my group, who developed this dunnart model in my group, he has a couple of papers in PNAS that may be of interest, because he's very interested in the evolution of different fibre tracts. So in his first paper, he identified that basically this anterior commissure is a corpus callosum with an anterior commissure sitting on top by doing a lot of mapping studies in the fat-tailed Dunnart and also looking at the platypus. And then more recently, together with Laura Fenlon, who's another postdoc in the group, and Annalisa Paolino, they actually started to do some misexpression studies in the Dunnart, looking at SATB2 and CTIP2. And so for those in the audience, CTIP2 is a transcription factor that's very important for descending pathways, and SATB2 is a transcription factor that's very important for formation of commissural projection, so the corpus callosum. And when you knock out SATB2 in mice, the corpus callosum doesn't form. Now, so what they were really interested, like what happens in a marsupial that doesn't have a corpus callosum. So it turns out that SATB2 is expressed in marsupials, and in fact, when they looked at using retrograde labeling together with immunohistochemistry, they can identify that SATB2 is expressed preferentially on commissural axons. But it had also already been shown in mice that there is a population of SATB2 and CTIP2 positive axons that project ventrally as well. So what they've done is overexpress SATB2 and CTIP2, and they've also used CRISPR in the Dunnart, because of course we don't have transgenic Dunnarts, but we can do in-pouch electroporation on them. And suffice it to say that, in fact, CTIP2 and SATB2 still play this role in medial versus lateral targeting, even in the Dunnart. And so those mechanisms do seem to be conserved, but they actually haven't tried overexpressing genes at the midline to form a fused interhemispheric midline. So that paper that I discussed was really just a descriptive paper looking at this fusion event in marsupials and monotremes. We didn't manipulate the expression of those genes in that. But what it does demonstrate is that you not only have to have the molecules that regulate medial targeting, but you also have to have the substrates. And so the midline glia are required for every commissure throughout the brain. Every decussation and commissure has glia associated with it, be it the floor plate or even within the optic nerves where they're crossing, or work from Carol Mason's group. Yeah, glia are really important for wiring the brain.
And have you run these Dunnarts in tests that perhaps they have lost something that the mice have gained by virtue of having a corpus callosum?
Yeah, so in behavioural tests, you mean?
Yes.
So we have not, and that is exactly what I want to do in my new lab at WashU.
Cool.
Yeah.
Thank you very much. That was a fantastic talk.
Oh, well, thank you for tuning in.
Linda, there is one more question if you have some more time. Okay. This is from Michelle Borde from School of Medicine here in Uruguay. Michelle says, I have a connection problems. Great talk. Thank you very much. I have a question. There is significant individual variability in central nervous system connectivity in many species, even when the functional outcome is relatively constant. To what extent do the reported alterations in connectivity explain the associated functional alterations by themselves?
Yeah, I think this is the crux of the most interesting question that we're trying to understand. So really, is it that you can have multiple different configurations of brain wiring in the brain that subserve exactly the same function? And I think that is absolutely a real possibility. But of course, to test that, we have to have good ways of actually identifying those changes in connectivity, and then very precise behavioural and cognitive tests that can pick apart those differences. So it's absolutely the case that at extreme ends, that, for example, conditions like microcephaly absolutely always result in intellectual disability, and changes in IQ and cognitive function. So at the extremes, there's definitely, you know, where things are more severe, there's definitely massive alterations. So brain structure is required for brain function, but to what degree? And I think it's a very fascinating question, because it gets at the crux of overall brain plasticity. You know, how is it that people can recover from strokes or other? Is it because they happen to have quite a diverse connectivity that was first set up, and now they can utilize those connections that weren't used before? I mean, that's complete speculation, but exactly how, and I think it's a question to test. It is really, you know, I don't come down one way or the other on this. I really want to understand what degree of connectivity is required for different cognitive processes. Good question.
Okay.
No answer.
Thank you very much, says Michelle Bordway, for your clear and very exciting answer. Okay, well, Linda, thank you very much for being with us. Now that you're moving to United States, maybe it will be easier for next Brain and Mind Evolution course that you, even you came last year from Australia, so next year it will be easier, even easier.
Yeah.
So thank you very much again, and thank you for all the audience to be with us. And well, keep in touch.
Yeah, look, thank you very much, everybody. I so enjoyed my visit to Uruguay, and I'm looking forward to seeing you all again. And please stay safe in this world of Zoom meetings. And
Yes.
thanks also to Trinidad for the interpretation. It was fantastic and very exciting to have a talk actually also translated into Spanish. Thank you so much.
Thank you so much. Thank you, everybody. See you. Bye-bye. Take care.